Thank you, folks, for joining us this gloomy but still a joyous occasion on Saturday morning. Welcome to our cancer screening in the Black community. My name is Delaney Hines, and my preferred pronouns are he, him. I serve as a member of the board of directors at SCAGO, as well as the education chair, overseeing much of the Learning for Life sessions that we brought to you over the course of this year. And I will also be fortunately serving as moderator for today's session. So if you or members of your network would like to refer to the information shared in this session at a later date, recording, as you can see, we're recording right now, of today's session will be posted on our website for viewing at your convenience. It's important for me to help you also articulate that one of our missions at SCAGO is to serve as a conduit for members like yourselves, members of our community, to access evidence-based, up-to-date information like we have prepared for you this morning. Today's session is important because barriers to cancer screening and racial disparities in cancer outcomes exist in Black communities, amongst others. So we hope to guide you through an engaging session that will equip you with the insight to better combat those barriers. As you can see, I'm joined by my fabulous guest, Dr. Juliet Daniel, who I'll be introducing shortly. But before providing an introduction to Dr. Daniel, I want to let you know that our agenda will start with our land acknowledgement and some additional housekeeping rules, which will then be followed by a presentation from Dr. Daniel and a Q&A session where you can ask any questions you might have. So with that, I would like to begin by acknowledging the Indigenous peoples of all the lands that we are on today. While we meet today on a virtual platform, I would like to take a moment to acknowledge the importance of the land which we all call home. We do this to reaffirm our commitment and responsibility in improving relationships between nations and to improving our own understanding of local Indigenous peoples and their cultures. From coast to coast to coast, we acknowledge the ancestral and unceded territory of all the Inuit, Métis, and First Nations people that call these lands home. I personally am joining you all today from the traditional territory of the Huron-Wendat, Anishinaabe, Haudenosaunee, and Mississaugas Islands, adjacent to the Algonquin First Nation, covered by the Williams Treaty. So we also ask that you save your questions, thoughts, or concerns until the latter portion of our session, where Dr. Daniel will be able to answer questions individually. As you know, this is a session that's currently being recorded, so please feel free to disconnect in the event you don't want to be recorded, but the session recording has begun. We kindly ask that you respect the learning being done in this space by abiding by the following housekeeping rules. If you could please keep your microphones unmuted, you'll notice they've been disabled for the duration of the webinar. If you'd like to ask a question, please do so by using the chat or Q&A function. I can also remove the mute disability and also allow you to orate your question during the Q&A. Just raise your hand. Closed captioning is provided, as some of you already noticed. And please use respectful language in the chat, so no profanity or inappropriate images or posts, please. Please do not post other material unrelated to today's discussion topic in the chat. And also please keep discussions and comments on topic as much as possible. SCAGO does reserve the right to pause a session or to seek removal of participants who do not abide by these rules. But once again, we thank you for your understanding and agreement to positive participation. Further disclaimer, remember that the information shared today does not constitute medical advice and is being shared with you for information purposes only. So no materials shared with you today are intended to be a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified healthcare provider with any questions you may have regarding a medical condition or treatment and before undertaking a new healthcare regimen. So please do not disregard professional medical advice or delay seeking it because something you was heard or said during today's webinar. So I know that was a lot, but without further ado, I want to introduce Professor Juliet Daniel, who is a cancer biologist and Associate Dean of Research and External Relations in the Faculty of Science at McMaster University. She received her Bachelor of Science from Queen's University, her PhD from the University of British Columbia, and completed postdoctoral studies at St. Jude Children's Research Hospital and Vanderbilt University in Tennessee. Dr. Daniel's cancer biology research led to her discovery and naming of a new gene, Kaiso, coined from her favorite Caribbean music, Calypso. Her research team is focused on elucidating the role of Kaiso in cancer and vertebrate development. Her team is also currently elucidating the molecular and genetic causes of the disparities in incidence and poor outcomes of triple negative breast cancer, TNBC, in Black women. Dr. Daniel co-founded the African Caribbean Faculty Association of McMaster, ACFAM, in 2010, and serves on the President's Advisory Committee for Building an Inclusive Community at McMaster. Professor Daniel also serves as a faculty member for student members of Women in Science and Engineering, WISE, and Black Aspiring Physicians of McMaster, BAPMAC. In recognition of her research and community service, Professor Daniel has received several awards, including the inaugural Canadian Cancer Society Inclusive Excellence Award, an honorary Doctorate of Science from the University of West Indies, UE, Cave Hill, a UE Vice Chancellor's Award, a WXN Canada's Most Powerful Women Top 100 Award, a 100 Accomplished Black Canadian, ABC, Women Award, and Barbados National Honor Gold Crown of Merit, among many others. She's also been featured in Millennium Minds, 100 Black Canadians, and Who's Who in Black Canada. So we are very, very fortunate and honored to have you joining us this morning, Dr. Daniel. So I will not take up any more of our audience members' time and pass it over to you.
Good morning, Delaney, and thank you and the board of directors and the organizing committee for inviting me to share and to do a presentation today about cancer in the Black community. All right, so good morning, everyone. I am not in Toronto, so I will give a land acknowledgement for where I'm speaking from. So I'm speaking from the traditional territories of the Mississauga and Haudenosaunee nations, and within the lands protected by the Dish with One Spoon Wampum Agreement here in Hamilton. And this Dish with One Spoon Wampum Agreement reminds or asks of each of us to take what we need from the land and leave some behind for others. So I'm gonna begin by just giving a simple overview of cancer. I'm not sure how many attendees are biologists or have any background in biology or the sciences. So cancer is actually a genetic disease, and it can occur in any cell in our bodies. However, there are certain tissues that it occurs in more often than others, in part because those tissues tend to undergo cell proliferation more frequently throughout our lifetime than other tissues. Cancer is basically uncontrolled cell proliferation, and then the spread of those uncontrollably growing abnormal cells to vital organs such as our lungs or liver or brain. In young animals, babies, cell proliferation actually exceeds cell death, which is why a baby grows from that 6 or 7 or 8 pounds that it's born at to an adult. In adults, we don't keep growing because there's now this equilibrium between the amount of cell proliferation and regulated cell death in our bodies to ensure that we don't become these giant human beings. However, in cancer, the tumor cells do not respond to that normal regulation that's happening throughout our body. And this is why the cell, once it's mutated, begins to then grow and grow and multiply and make more cells. There are many different types of cancer, and most of them are actually named after the body part they begin in. And so this is a little simple schematic showing you some of these more common cancers. So many of you have heard of lung cancer, breast cancer, colon cancer, bladder cancer, prostate cancer. And of course, what we call the liquid cancers, leukemias, and lymphomas. And then there's some other types of cancers called sarcomas, because those tend to be in unusual tissues such as the fat, the bone, and the muscle. So what actually causes cancer other than this uncontrolled cell growth? So it's actually due to a series of mutations that occurs within the DNA of our cells. These mutations in the DNA are actually caused by internal factors. So these could be metabolic products of things that we've consumed. So we've either ingested or inhaled, or external factors, so chemicals that we're putting on our skin, etc., sunlight, cigarette smoke, stuff, factors like that. So these will be called carcinogens. So this little simplistic dramatic diagram at the bottom here, what I've tried to show is that UV irradiation, cigarette smoke, for example, can induce these mutations shown here by the lightning bolt. This can cause just one change in the DNA, but that one change could impact whether or not a protein is made the correct way and whether or not it will function normally or abnormally. And so it's usually due to multiple mutations in different genes within the same cell that ultimately causes cancer. Random mutations will not necessarily cause cancer. It's usually a very specific type of mutation within the gene. So in the mid-80s, there was this discovery that we all carry the same genes, as you hopefully know, but there are genes in our DNA that normally encode proteins that are healthy for us in healthy individuals. And these genes were classified into two different types. There were oncogenes and tumor suppressors. Oncogenes are any gene that encodes a protein that can cause tumors in an animal. We all have these genes, but when they're behaving properly, we call them proto-oncogenes or pro-oncogenes. We all have them. Tumor suppressors, we all have as well. And tumor suppressors, as the name suggests, encode genes that encode proteins that prevent cell proliferation. So cancer, or what we call the molecular basis, so within the cell, what is actually causing cancer, we call this the molecular basis of cancer, and it's due to increased oncogene activity. So now you've got lots of proteins that can induce tumors or cell proliferation and decrease tumor suppressor activity. And I like to think of this as a traffic light. Your stop sign, which is your tumor suppressor, is usually serving a great purpose at an intersection, is preventing us from colliding and crashing into each other. And when you get that lovely green sign, green light, it's the sign to go, everything's safe and is clear. So you can imagine if we were at an intersection where our stop sign was no longer working and every single traffic light was green, we would have complete chaos at that intersection. And that would be due to increased oncogene activity in this case and decreased tumor suppressor or red light activity. So this is a schematic of what it would actually look like in a mammalian cell, or mammals, I should say, humans. So our normal cells are dividing happily. As I said, there's an equilibrium in all of us. For example, the cells, our skin and our nails are constantly proliferating and shedding, and our hair. Some of us more than others, but that's just the way we're all designed. And normally there's this equilibrium between cells proliferating and dividing. However, occasionally a cell will acquire a mutation that may lead to cancer. But our bodies also have a process to detect that cell and to eliminate it to ensure that it doesn't cause harm to our bodies. And this is a process known as apoptosis, or programmed cell death, or cell suicide. However, in cancers, what happened is this cell evades that surveillance to eliminate it, and it starts to multiply and produce way more cells. So suddenly it doesn't undergo cell death or suicide, but it starts to grow uncontrollably. So now we have, several cells with mutations. They each multiply. And as they multiply, they acquire more mutations. And the more mutations they now have this unfair advantage over the healthy cells, because now they can keep proliferating uncontrollably. Our bodies can't keep up to eliminate them. And ultimately, they give rise to this mass or this clump of tumor cells that are then usually visible or palpable, whether it's visible on an MRI or a CAT scan. And in some cases, we only realize we have cancer because it manifests as other symptoms in our bodies. So for example, ovarian and pancreatic cancers are typically only detected or diagnosed because they trigger other symptoms in our body that then lead to the physician requiring or requesting various tests to determine what is causing the symptoms. So every year, the Canadian Cancer Society releases updated statistics and data on the incidence and prevalence of cancer in Canada. So every year, or at the moment, I should say, we have over 1.5 million people in Canada living with and beyond. And when we say beyond, we mean that they currently are what we would consider to be completely in remission with no cancer in their body, but they are counted in this 1.5 million people who were basically at some point diagnosed with cancer, and then there are those and are no longer and are considered cancer-free, and they're combined with those who were recently diagnosed with cancer and are undergoing treatment, etc. So if we look at this pie chart on the right, you'll see that this prevalence for all the different cancers. So the majority of the people currently living with or recently diagnosed with breast cancer is one of the largest groups of people in Canada, among that 1.5 million, followed by men with prostate cancer, and then various other cancers, colorectal, melanoma, etc. Many of you will be surprised to see that lung cancer here is very small, but this is because the prevalence rate is the combination of the incidence of a type of cancer and the survival. As you know, lung cancer has a very poor survival rate. So the reason this is only 4.1 is because many lung cancer patients are not surviving, and therefore this pretty much represents those that are more newly diagnosed than those that are, including all that have been diagnosed in the past 25 years. So this is all over a 25-year period. So, oh, I forgot to mention this. So what this also means is that the increased incidence and longer survival for many Canadians, not just in Canada, actually for many people globally. So this increased incidence in cancer coupled with longer survival times means that many of us are living with cancer or beyond cancer, but it also means that this could be a burden to our healthcare system because now we're all living longer, and we have what they would call these comorbidities that require us to seek medical care more often. If we break apart that pie chart by sex, so you'll see that in the case of men, the highest prevalence of, not surprisingly, is prostate cancer. For women, it's breast cancer. And again, lung is small again because patients diagnosed with lung cancer don't tend to have long 25-year survival periods. So what's really interesting is, like, to show this diagram is that unfortunately, globally and definitely in Canada, we do not collect a lot of race-based data. So a lot of the data and statistics that's out there regarding cancer incidence and mortality is based on white or Caucasian populations, or white or Caucasian people that respond to many of the surveys. So one of the really interesting findings recently is that globally, the top three cancer incidents in the world are breast, lung, and colorectal cancer. However, for women, in the Caribbean, it is breast, cervical cancer, and colorectal cancer. If we look at the top three causes of death globally, it's lung, breast, and colon cancer. But in the Caribbean, it is breast, cervix, and and colon cancer. So you can see how, if we're trying to, think about this, or we hear these things on the news and we think it doesn't impact us or it's not relevant to us, it's slightly problematic because we were not necessarily included in the analysis and the data that generated these more general findings. Similarly, for men, the highest incidence of cancers globally in men is prostate, lung, and colon cancer. In Caribbean men, it's the same, prostate, lung, and colon cancer. However, when you look at the highest incidence of death via cancer, in the Caribbean, it's prostate cancer is the #1 cancer among cause of cancer death among men, compared to globally where it's lung cancer. And you'll notice as well that globally prostate is #3, but in the Caribbean and Black men, it's prostate cancer deaths are #1. So this is one of the reasons why myself and many others that do cancer biology or that are physicians are very concerned, because there are clear differences in the incidence of various cancers in Black populations, as well as clear differences in the death rates from some of these cancers. So how did I begin doing cancer research? So I was born and raised in Barbados, was always fascinated with the human body and the natural world. One of those kids that was always asking, why is the sky blue? Why are there so many different types of animals? Why are they, I don't know if any of you ever had this question, like, we have very few blue flowers on the planet, right? Most flowers tend to be red, yellow, orange, like we just don't have a lot of blue flowers. And that was something that always puzzled me. So anyway, I used to ask lots of questions and I was a real bookworm. I really excelled at math and physics. But somehow in the Caribbean, girls were not really encouraged, or no one ever thought about a girl being an engineer, which would be the logical thing to recommend to a girl that's good at math and physics, but it was never recommended to me. I grew up in a low-income family, so no one in my family could advise or knew what I should do. As far as they were concerned, I was good at math and physics. Math and physics are sciences. Sciences means you should be a physician. So I was encouraged to be a doctor. I did know, however, that I wanted to do research with global impact. So my parents saved as much as they could, made lots and lots of sacrifices, and saved enough for me to come to Canada University for my first year. They literally saved enough for the first year only, and we were going to figure it out 1 year at a time, which we did. So I went to Queen's University and had a wonderful time. And the reason I chose Queen's was because they saw me as an individual and not just an applicant. All the other universities I applied to did not acknowledge, they acknowledged my application, but that was the only communication I got. Queen's sent me, every 2 weeks I got something from Queen's before I submitted my official acceptance. And so when the time came to decide, my family and I thought I should go to Queen's because we at least knew what we had. They had sent us pictures of Kingston, pictures of Canada, maps of Kingston, copies of the local newspaper, etc. I had a wonderful time at Queen's. However, my last year at Queen's, there was a series of unfortunate events, as I call it, serendipity, destiny, I'm not sure. But basically, my neighbor in Barbados died from breast cancer. And then a month after she passed away, my mom was diagnosed with ovarian cancer. And that same semester, one of the professors there, Dr. Lita Raptis, came to class. She was a new professor herself. And I remember her coming to class and being super excited by the discovery of those genes that I told you about, this class of genes called oncogenes. And she was convinced, like every other cancer biologist in the mid-80s, that the discovery of these genes would lead to the cures for cancer in 30 years. And sadly, we're beyond the 30-year period and we haven't fully cured cancer, but we have increased the survival rates from cancer. Unfortunately, my mom passed away from ovarian cancer 6 months after her diagnosis and 4 days before my convocation from Queen's. So cancer chose me, I say, because I didn't necessarily chose cancer research as a career at that point. I hadn't decided what I wanted to do. However, the novelty of snow and cold had worn off at this point. Everyone told me that UBC and Vancouver was the warmest place in Canada. There was no Wikipedia or Google at that time. So I just believed everyone, looked at it on a map, applied, got accepted. And after spending the summer in Barbados after my mom's death, I moved to Vancouver. I spent 6 years in Vancouver doing my PhD, but it was what we would call fundamental research. I wasn't doing any research really that was directly applicable to human cancer treatments or cures. And I wanted to do something that was way more applicable to human cancer. And the one hallmark of cancer that I had learned about during graduate school was this hallmark of cancer known as the activation of invasion and metastasis. Basically that process where the tumor cell breaks away from the primary site in the breast or the prostate and spreads to the brain and the lung and the liver, which ultimately causes the death and the mortality of the patient. There were all these other hallmarks as well, as I said, like tumor cells grow uncontrollably, they constantly proliferate, they can somehow evade and avoid tumor suppressors and resist cell death. But I really wanted to understand what made them break away from the tumor and spread, because I felt that that would be a vital part of this whole puzzle to try to solve. So I made the rather mature move at the time. I had job offers in San Francisco and London, England. But if I went to those labs, I was going to be studying one of those oncogenes that had been discovered, 20 years earlier, and where many, many people were studying those genes already. And I made the decision to go to Memphis, to St. Jude Children's Research Hospital, to work with a new professor there, Albert Reynolds, who had just started his lab 2 to 3 years before I applied to his lab. And he had basically identified a new protein that he named P120-catenin that was a substrate of one of those first oncogenes, the SRC oncogene. And he was the only person in the world at the time studying this protein P120 and its role in cell-cell adhesion. Now, most metastatic tumors have defects in cell-cell adhesion. And so this to me was the perfect opportunity to really understand why tumor cells break away from the primary tumor mass and spread to other organs. Unfortunately, Al died unexpectedly 4 weeks ago at the young age of 66, and many of us are still grappling and coping with that, with his loss. But basically my project when I joined his lab was to try to determine and understand the role of P120 in the cell adhesion complex that was known to malfunction in 50% of human tumors. So my project was, what is P120 doing in this process? I utilized a technique to identify other proteins that interacted with P120 in the hopes of understanding what P120 did. And the reason we do this is like anything else. If you're trying to study in science, if you're trying to study something, there's two ways to do it. You either make a lot of it and see how it impacts the cell or the organism, or you remove it and see what happens, or you look at other proteins that it's interacting with. So I did an assay to identify proteins it interacted with, and the gene most frequently identified encoded a novel gene. It was a novel gene, I should say, and Al said I could name it anything I wanted. Being from the Caribbean, being to numerous conferences by that time and seeing very few Black scientists, I wanted to name it something Caribbean, and I opted to name it Calypso after our Caribbean roots. And because Calypso is my absolute favorite type of music, but I didn't want to call it the Calypso gene, so I wanted to give it something a little bit more subtle that, you know, mainly only Caribbean people would know. Turns out kaizo is also a Japanese word meaning salad, but that's okay. That's a healthy, good thing. So what is kaizo? It's a transcription factor. Unlike most transcription factors that recognize one specific region of DNA, kaizo recognizes two types of regions on DNA. It recognizes what we call these methylated sites on DNA, and it also recognizes specific DNA sequences. It regulates many biological processes, and interestingly, most of these biological processes play roles in tumorigenesis and cancer progression. So it regulates genes that control cell proliferation, apoptosis or cell suicide that I mentioned earlier. It regulates genes that play a role in cell motility, which may explain how the tumors leave the primary tumor and move to other parts of the body. And more recently, we found that it also regulates genes that play a role in inflammation. And we're also now learning that constant inflammation within our body could also be a risk factor for cancer formation. And we know from other studies and other researchers around the world that kaizo plays roles in multiple cancers. In my lab, we're only studying kaizo in breast and colon cancer, but there are other labs around the world studying kaizo in prostate cancer, pancreatic cancer, and lung cancer, among others. So I would say the first 8 to 10 years at McMaster, I was just characterizing kaizo. What does it do? How does it do it? And then in 2008, I went to a conference in the US, the American Association for Cancer Research, or AACR conference. And at that conference, I heard a breast oncologist talk about the work she was doing in West Africa. So she was going to West Africa and teaching the surgeons there how to perform breast mastectomies and breast surgeries, lumpectomies, mastectomies. And she was observing this really, really high incidence of a breast cancer subtype that she was calling triple negative breast cancer. I had never heard about this. I had already started planning my retirement very early at McMaster. They encouraged us to do this. And I knew that I wanted to give back to the Caribbean and ultimately also, if possible, conduct research in the Caribbean. So what is triple negative breast cancer? So all breast cancers are categorized or subclassified based on the expression of 3 proteins on the cell. There are 3 proteins many of you may have heard of: estrogen receptor, or ER, progesterone receptor, PR, and the HER2 receptor, or HER2. And when a patient is diagnosed with breast cancer, the tissue, the biopsy tissue, as well as the tissue removed during the lumpectomy or mastectomy, is screened and analyzed for the expression of these 3 biomarkers. Breast tumors expressing the ER protein can be treated with the drug known as tamoxifen. Breast tumors expressing the HER2 protein can be treated with drugs known as Herceptin. However, as the name triple negative breast cancer indicates, patients diagnosed with TNBC do not express ER, PR, or HER2. Consequently, there are no targeted therapies that can treat triple negative breast cancer patients. They can only be treated with radiation therapy and classic or standard chemotherapy. But what was really interesting is that for some reason, triple negative breast cancer has the highest prevalence in young premenopausal Black and Hispanic women compared to other women. And it also has an incredibly poor prognosis. It's very aggressive and metastatic. And this is in part due to the lack of specific therapies. Most of the data on triple negative breast cancer, when I first heard about this in 2008 and started doing some reading of the literature, was based on data from the US, because regrettably, Canada and many countries around the world do not collect race-based or ethnic data on any disease whatsoever. And the US, despite all of their foibles, they have been collecting race-based data on every single disease since the 1970s. So they have a wealth of information on the incidence and outcomes of most diseases based on gender, based on ethnicity. It's an incredible source of data. So the data in the US was suggesting that perhaps this high incidence and poor outcomes in Black women in the US was due to their socioeconomic status and limited access to healthcare, because that's how their healthcare system works. However, while that was explaining the poor health and disease outcomes, it wasn't necessarily explaining why the incidence was higher in Black women to begin with. A few years ago, the World Health Organization published their annual report, and they've stated that breast cancer is actually an emerging health crisis in Africa, because this 5-year survival rate is less than 50% compared to more than 90% in North America, including Canada. And so it might surpass, for example, the death from HIV and malaria if we don't address and try to understand why this triple negative breast cancer has such a high prevalence in young Black premenopausal women. This graph, again, from the US data, shows the incidence of breast cancer in white women indicated in the green line and in Black women indicated in the indigo or blue line. And you'll see that historically, the incidence of breast cancer has always been highest in white women, in part, we think, just because of more awareness and self-advocacy compared to other women of other ethnic groups. What's really interesting, however, the graph on the right shows the mortality rate from breast cancer. And what you'll notice is that the mortality rate from breast cancer among white women started to drop precipitously in the early 1990s and is now relatively low compared to where it was in the 1970s. And this is what's contributing to that 90% survival rate that I just mentioned for breast cancer in North America. What you might also notice, however, is that the indigo or blue line, which represents breast cancer mortalities among Black women, kept increasing in the 90s and then only started to gradually decrease. But you'll notice there's still this significant gap in the mortality rate between Black women and white women and women of other ethnicities. And many of us now think that this big gap is due to the fact that Black women are more frequently diagnosed with triple negative breast cancer, for which there are no known therapies. And interestingly, most white women are diagnosed with estrogen receptor positive tumors, for which we have a specific therapy, tamoxifen. So this, we think, is contributing to this disparity in outcomes and survival among Black versus white women, not just access to healthcare, but genuinely just the biological makeup and difference of the cancers that we tend to impact us more. So when I started, as I said, I was fascinated by this, and for my first research leave in 2011, I actually went to Barbados and started collaborating with physicians there to try to see whether or not triple negative was also a problem in Barbados and the Caribbean. Around that same time, I got a new graduate student, Blessing Bassi Arkabong, who was Nigerian, and she joined my lab, and we started to look into this in more detail. At the time, I was also collaborating with researchers in the Netherlands who were also studying P120 and cell-cell adhesion and its role in tumor metastasis. And Dr. Dirksen and I published a paper in 2012 where we reported that high Kysol levels were found in invasive ductal breast carcinomas, but not in invasive lobular carcinomas, suggesting that these high levels of expression of Kysol could be contributing to the poor outcomes in patients with invasive ductal carcinomas. Blessing then turned to the internet and started to peruse all of the data that's publicly available regarding the survival rates of all women diagnosed with breast cancer in the US. As I said, a lot of this is US data. And she looked at the Kaizo expression levels in those patients. And what she found here is that patients that had very high levels of Kaizo expression had very poor survival. So the axis on the left is showing you the survival rates. And so you see that the red, it has the lowest survival rate compared to those with the blue line, and the blue line are patients that had very little Kaizo expressed, and they were living longer than people with lots of Kaizo. So we hypothesized that this high Kaizo expression was in some way contributing to that metastatic spreading phenotype of the breast tumors, which were then leading to higher mortality rates of the patients. So what Blessing did next was to take some triple negative breast cancer cells that we have in the lab that many people around the world are using to study triple negative breast cancer. This cell line is known as MDA231. She took this cell line, as I said, which is known to be very aggressive and metastatic in mouse models. And these are, I'm not going to get too technical, but basically these dark blobs or lines represent the amount of protein in the cells. So you'll see that the parental MDA231 cells do not express any of the cell adhesion protein E-cadherin that keeps all of our epithelial cells stuck together. However, they express lots of Kaizo. So Blessing made 2 clones from this cell where she knocked down or decreased the expression of Kaizo. So you can see here, she's eliminated the expression of Kaizo. And surprisingly, what we saw was that when she knocked down Kaizo, we got an increase or re-expression of E-cadherin. And this is consistent with what we saw when we looked at the cells under the microscope. The cells went from having these kind of elongated, spindly appearance, what we would call fibroblastic or mesenchymal, to suddenly having these more flat cuboidal appearance. And you can see that the cells are touching each other much nicer than they are in the image on the left. So these cells have undergone what we call a mesenchymal to epithelial transition. And this is great because it means that these cells on the right with very little Kaizo are not going to move very far. So this leads to decreased cell motility. So this is all, however, on a plate in the lab. So then we wanted to see whether what would happen if we went into an in vivo model and we used the mouse for this study. So what Blessing did is she took 2 batches of mice that are immunocompromised and would allow us to inject these human TMB cells without kicking them out of the mice. So the mice were injected with the MDA231 cells, the MDA231 cells that had a lot of, sorry, the sun came out in Hamilton, with the MDA231 cells that had lots of Kaizo. And then she took another batch and injected them with the cells that we had removed or knocked out most of Kaizo. And in this case, I've just indicated Kaizo by the letter K. We then waited 8 to 12 weeks and we kept monitoring the mice, and you can actually see the tumors growing bigger in the breast region of the mice. And then once they had grown to a specific size, we sacrificed the mice and then looked at the lungs and the liver of the mice to see if any of the human cells that we had injected into the breast had somehow made their way to the lung and the liver. And this next diagram shows you that this was a great experiment, at least from a scientific point of view. Hopefully you can appreciate the importance of this. So on the left are the parental MDA231 cells. The mice injected with the MDA231 cells, and these are images of the lung and the liver from those cells. The dark purple regions of the, oh my goodness, why is it doing this? The dark purple regions represent the human TMB cells that are now proliferating in the lungs and the liver of those mice. This was an experiment everyone knew. This is traditionally what's known about MDA231 cells. Many scientists around the world knew that it was an aggressive cell line. It always spreads to the lung and liver. So this is what we would call our control. This was what we expected. We didn't know, however, what would happen when we injected cells where we had depleted or knocked down Kaizo. And I hope you can see there's a huge difference between the mice injected with the kaizo-depleted cells versus those injected with the TNBC parental cells. And I hope you can see that the mice injected with kaizo-depleted cells have very, very little or few metastases compared to the parental cells, both in the lungs and the liver, indicating that in this scenario, the simple removal of just kaizo, we didn't do anything else to the cell, caused or prevented the spread of the tumor from the breast to the lung and the liver. The mice still had tumors in the breast, they just did not spread to the lungs or the liver. So we have many ongoing experiments now to try to obviously figure out mechanistically what's happening, and I don't have time to go into all that detail, but it's a really exciting time in the lab right now. So as I mentioned, this all started when I heard Dr. Lisa Newman talk about the incidence of triple-negative breast cancer in West Africa. And the incidence of TNBC actually varies across Africa. In East Africa, it's quite low. It's 15% to 20%. But in West Africa, it's as high as 60 to 80% in Nigeria and Ghana. And most of you know that many Blacks in the African diaspora or globally originated from West African slaves during the transatlantic slave trade. In the Caribbean, the incidence of TNBC is 15 to 20%. And in North American Blacks, it's also about 20%. And as I said, among white women, it's half that, it's about 10%. So we postulated or hypothesized that perhaps there's an ancestral predisposition from our African ancestors that's leading to this high incidence of triple-negative breast cancer in North American and Western Black people. So because Blessing was from Nigeria, we also established a collaboration with physicians in Nigeria. I collaborated with physicians in Barbados, and then some Jamaican colleagues heard about our studies and wanted to get involved. So now we have breast cancer tissues from women in 3 countries that are ancestrally related through the slave trade. And our question that we asked was this kaizo expression levels correlating with patient survival in these populations. So what we did was we took the TNBC tissues that we had, we made a pilot tissue microarray, which is an assay that allows us to take a small piece of tissue from each of the biopsies taken at the hospitals in these countries. And instead of screening each patient's tissue one at a time, we took a small piece of tissue, embedded it into a wax block, which is shown here. So these little round dots represent a piece of tissue from individual women. So this allows us to screen many, many tissues from many women at the same time, rather than trying to screen 100 women one at a time. And this is what it would look like when we stain it with antibodies to the proteins that we're interested in. In our case, we were looking at the expression of kaizo, as well as the established breast cancer predisposition gene BRCA1 that many of you may have also heard of. The brown staining indicates protein expression, and the darker the brown, the more protein there is. And I hope you can see in this slide that women with high African levels of African ancestry, so Nigerian and Barbadian, had way more kaizo expression in the nucleus, which are the dark brown circular areas, and the cytoplasm, which is slightly lighter brown, compared to African American women and compared to white or Caucasian women. And these graphs on the bottom just show you that this was a statistically significant difference. So the amount of nuclear staining of expression of kaizo was significantly higher in Nigerian women compared to white women. It's also higher in Barbadian women compared to white women. And although the African American women, it's slightly higher than white women, it was still significant, even though it's not that different if you look at it on the graph. So again, this actually suggested that the higher levels of kaizo are occurring in Black women, and perhaps this is contributing to that disparities that we're seeing in Black women incidence and survival rates. At the same time as we were doing that, we were also doing a parallel study where we were actually sequencing the DNA of the women in our study. And we were trying to see whether or not there were specific genes or mutations that were causing this high incidence of breast cancer, or that might explain why they have poor survival. You don't need to try and understand this. This is just to show you that we took the tissues, we sequenced the DNA, we're looking basically for mutations that might explain why we're getting more triple negative breast cancer than other women, or why we have poor outcomes to triple negative. This Venn diagram on the right, I know it might bring back bad memories for all of you that hated math and calculus, etc. But basically, this allows us to identify which genes are common or unique, common to all women. So this is white women, Barbadian women, Nigerian, African American. And so the genes that are common to all four of those populations are these here in the middle, the 78 genes. And then this Venn diagram also allows us to see that there are 94 unique genes only to Barbadian women, 72 unique just to Nigerian women, and over 2,000 unique to white women, and 300 unique to African American women. So this is a lot of data for us to go through now and to try and analyze, but we will be starting likely with these 78 in the middle, because these genes could help us develop or identify a gene that's contributing to triple negative breast cancer in all women. And then these 94 and 72 might help us understand what's contributing to the higher TNBC in Black women, and these, especially these 56, which are overlapping between Barbados and Nigeria. So I'm about to wrap up shortly, but I also want to point out that although in my lab, as I said, we're doing mainly breast and colon cancer research, and I didn't show you any of the colon cancer research in the interest of time, I want to point out that there, one of our collaborators in the States, in the US, has been studying Kaizo in prostate cancer. And what they found is that high Kaizo expression correlates with prostate cancer aggressiveness and poor survival in Black men. So this is very similar to what we're finding with breast cancer in Black women. High Kaizo, lots of aggressiveness, poor survival in breast cancer. And now he's finding high Kaizo, very aggressive prostate tumors and poor survival in Black men, suggesting that this high expression of Kaizo in Black populations may be contributing to aggressive tumors. And maybe we can try to target or at least screen for the expression of Kaizo as a risk factor, so that people can determine whether or not, how they can change their lifestyle and or what preventative measures they can use to have a longer life. So back to breast cancer for a little bit, just to mention for any women listening, there are several risk factors. So it could be or your age, your family history. Most breast cancers, however, are not due to family history. Maybe only 10 to 15% are because a close female relative has had breast cancer or ovarian cancer. Most of it is unfortunately just due to random chance and mutations. It could be hormonal changes. Some people are there's still a big debate as to whether or not oral contraceptives play any role whatsoever in breast cancer. But we do know that lifestyle, smoking, being overweight, not exercising, etc., plays a role in breast cancer development. And of course, we've got the typical environmental factors that affect all cancers. So what can you do to lower your risk of any cancer, not just breast or prostate, but any cancer, and in fact, any disease, is basically try to eat as healthy as you can. I know it's hard, especially with inflation right now and the high cost of living. Fruit and vegetables are among the most expensive things at the grocery store. And because of that, we tend not to buy those things. We tend to buy those cheaper, high carb, processed foods, which is not good for us in the long run. And so, and of course, we all know we should quit smoking, limit our sun exposure. We need to be physically fit. And as I was telling Delaney earlier, I was one of those people. Many of my friends and colleagues and people on Twitter, et cetera, during the pandemic were binge watching TV and baking cakes and baking bread and goodness knows what else. And everyone was like, did you watch this movie? Did you do this? And I was like, no. And I couldn't figure out what was I doing? How come I didn't make cakes and binge watch TV? I literally didn't do any of that. And then I realized I was walking 90 minutes to 2 hours every single day during the first year of the pandemic. So instead of watching TV or baking cakes and more carbs, I was actually out walking for 2 hours every single day. And not necessarily all in one block, but in between meetings, because we were all in Zoom meetings as well. And so I would encourage everyone to just get into that habit of being physically fit, because it makes a huge difference. And even if you are unfortunate enough to be diagnosed with cancer, being fit also helps with your recovery, as well as your chances of survival from that diagnosis. Ultimately as well, early detection is key. You have to know your body. You should know every lump and bump and ache and pain, but not be a hypochondriac, but just really be aware of changes in your body so that you can alert the physician, your family doctor, as soon as you can. So some of the recommendations that, many of us have for improving Black health, whether it be, is that, we need more Black health professionals. We do not have enough Black health professionals in Canada, and we do not even have enough Black cancer biologists in Canada. To my knowledge, there are only 3 or 4 Black cancer researchers across all of Canada. We have several Black students studying with us, but at the professor level, we do not have more, we have less than 10 Black cancer biologists in Canada. That is like, we need more Black researchers to tackle these problems and to ensure that we have improved health as well as improved and better treatments for some of these diseases, not just cancer. For example, sickle cell is a great example. We just don't have enough Black researchers. We need Black researchers to ensure the research will be done that specifically impacts our community. We need our health professionals to have better cultural awareness and sensitivity. I don't know how many of you know Dr. Anya Norum. She has been advocating for this before EDI and equity became trendy in the past 3 years. And so I know there's the Black Physicians of Ontario, and likewise, they have been advocating for many of these things before George Floyd was murdered. And obviously we need better screening programs and access to care. We also need Black people to participate in clinical trials. If we don't participate in clinical trials when these drugs are developed, they may not work on us because we did not participate in the clinical trials. And that leads back to why we need more Black health professionals and researchers, because many of us do not trust the healthcare system because we've been screwed before by the Tuskegee trials, etc. So ongoing and future studies in my lab determine why Kaiser is even highly expressed in these aggressive cancers to begin with. We have no idea. We're validating the clinical relevance of our findings that Kaiser is highly expressed in aggressive tumors to see whether or not we could develop Kaiser as a diagnostic or prognostic test to help Black men and women determine whether they're at risk for prostate and breast cancers that are aggressive. And we're, as I said, analyzing all that genomic data that we got to see if there are any novel mutations that might help us to further also develop diagnostic prognostic tests or new therapies for Black people. And as you know, we're wrapping up the International Decade for People of African Ancestry, ends in 2 years. So we really have to get ourselves in gear, and we should all be lobbying and advocating our local or municipal, provincial and federal governments to actually do something significant for Black Canadians during the next year and a half. Again, the US is incredibly good at collecting data and literally being at the forefront of this, even though they're also kind of a bit racist in certain areas. But one of the things that's coming out of research in the US is that they're now considering racism trauma as an actual social determinant of health. So not just poor, low education or low income or unemployment, but they're now thinking that just the act of racism and the trauma that causes could be causing these epigenetic or these changes in our DNA at the DNA level that is then contributing to our poor health outcomes. And I usually make this business case for anti-racism framework. It actually costs more as a society to address the consequences of racism. We saw this in the pandemic, where we had that vaccine hesitancy among Black, South Asians, Muslim. Many communities did not trust the healthcare system. They didn't want to take the vaccines because they thought it was some new thing to, eradicate all of us. And this just exacerbated then the health and the economic disparities among those populations. We also end up with late stage diagnosis of various diseases, whether it be diabetes or cancer, again, because there's no trust of the healthcare system, because we have very few healthcare workers who look like us, which then for every organization leads to lost wages, and then they have to pay these short and long-term disability costs. So I'm sure I don't need to preach to you guys here, But, you know, we all need to engage in transformative work. We need to collaborate with Black researchers and clinicians. We need to push to get our workforce diversified so that we have all voices at the table, because the more diverse we are, the more creative we will be in our solutions as well. And because there, like, as I said, there's only 3 or 4 cancer biologists, we're stretched thin. We're being consulted to fix a situation we didn't create, but we're trying to fix and help these organizations that created it. And anyway, that's another story. But yeah, and also partner with our Black-led health organizations. There's several, especially in Ontario, we have many. We have the Black Health Alliance, we have the Taibu Community Center, we've got the Olive Branch of Hope Cancer Support Service, and I partner with them on many cancer outreach initiatives over the past 7 years. And ultimately, you know, for those that are not Black, they need to acknowledge their privilege and their benefits and their successes, and that it, you know, that those benefits and privileges don't necessarily extend to everyone. And representation with agency, so it's not a matter of just hiring or recruiting new Black staff to your organization, but their voices need to be heard and valued. So I call that representation with agency. And again, I'd just like to end by acknowledging my mentor, Al, and friend. You know, it was in his lab that I cloned and discovered Kaizo and basically got my career established as a cancer biologist. And this was taken at, I think, probably his birthday party before I moved to Canada as a professor. And we were very tongue in cheek and gave him a gift in a Victoria's Secrets bag. If you knew him, he was very introverted. And yeah, so highly missed by many of us. And I just want to acknowledge all my collaborators globally and here in Canada. And we have a breast cancer fundraising team that we call Pirates of the Caribbean. And this was us at a walk in I don't remember now, 17 or 18. It was the 10th anniversary of the walk in Hamilton. And if there are any young scientists out there, I'm hiring. I'm looking for a Black postdoctoral fellow, research associate, or grad student. So please reach out to me. And I will end there and take any questions you have. And just put this picture up to brighten up your day since it's raining and the sun's trying to come out in Hamilton. Hopefully it is wherever you are as well. Thank you.
Thank you so much, Dr. Daniel. First and foremost, my heartfelt and deep condolences for the loss of your longtime friend and mentor, Dr. Reynolds. I'm sure he impacted many lives beyond your own, but happy he was able to at least, you're part of his lineage and can carry his intellect and thought process forward. So with that, I want to make sure I provide folks in the audience with an opportunity to ask any questions you may have of Dr. Daniel. Maybe while folks are thinking, I do have a question of my own, just wondering about what we can do from a public health standpoint to raise awareness of the Kaizo gene and its impact on, cancers and the proliferation of cancers. Is there anything we can advocate for in terms of screening, like you mentioned before?
Not necessarily for the Kaizo gene, but I think all any Black person working in public health should be working with the Black Physicians Association to make sure that they are somehow at the table meeting with, as I said, at the municipal and provincial and federal level, any minister of health, to point out that the current policies, even regarding screening, do not help those of us that are from the Black community. As I said, triple negative breast cancer impacts women in their 30s and 40s, but the age for mammography screening is 50. In the US, the age is 40, which is 10 years makes a huge difference. And again, triple negative breast cancer doesn't just affect Black women, it affects white women as well. And one of my dear friends, her sister-in-law's sister, is currently a young 35-year-old white woman in palliative care from breast cancer, leaving behind, and will be leaving behind 2 kids under the age of 7. Right? And I don't know how we can impress upon our governments and healthcare workers and our ministers of health that you cannot have breast cancer screening starting at 50 when there's a really aggressive breast cancer subtype impacting women in their 30s and 40s, when many of them aren't thinking they're going to get cancer. They're busy setting up their careers. They're busy starting a family. And, and if you're breastfeeding, you might feel a lump and think, oh, well, that's a breastfeeding issue, right? you're not thinking you're having cancer, because as well, the TV and the media never shows, first of all, they never show what young women getting breast cancer.
No.
And they almost always show just white women getting cancer.
Right.
So a young white woman as well is not going to think she's getting cancer, and definitely not a young Black woman is going to think she's going to have breast cancer. So I think that's where, like, from what I call the low-hanging fruit, is just getting all of our cancer agencies, the Canadian Cancer Society, the Breast Cancer Society, like, they need to start using commercials and ads that show the diversity and spectrum of people that get cancer, and to show that, yes, a 25-year-old can get breast cancer and can die from breast cancer. Because a lot of young people do not know that. And even though I have young people in my lab, when I talk about this, I think none of them think it will happen to them.
Right. Right.
Right? It's like, I don't have a family history. And I'm like, but you also know that only 15% of breast cancer patients have a family history. You were in the 80s, you have no family history, but you're in the larger proportion of 85% where it's going to be random. Like,
right. Yeah. You're at risk.
So a lot of it is, I think we need to be at that table regarding health policy changes. And also, and this goes back to the cultural sensitivity of our healthcare workers. There's a young Black lady that I met a month ago who found the lump. Granted, it was during the pandemic, so it was tougher to insist and get the necessary screening she needed. But again, she was told, oh, you're too young. That can't be a lump.
Right.
Like, she was not prioritized to even get the test to see if it was a lump, right? Or a tumor. Right. And so, because many physicians still have this idea that breast cancer happens to older people. It does not happen in young women, especially in their 30s. Like, you're 34? You're too young to have breast cancer. And I'm thinking, and because many of our doctors, although they are required to do these continuing medical education courses, I'm not sure that those courses are talking as much as they should about the changes in medicine over the past 30 years. Right? And so again, this is where I think that partnering with the Black Physicians of Ontario, and I don't know if every province has a similar association, but definitely in Ontario, we're super, super active. They're really advocating. I think just partnering with them, I don't think they don't need donations, but I think partner with them, because they're the ones really lobbying and advocating for changes in healthcare from the perspective of Black communities, but also Indigenous. Like, we really need our ministers of health to do better.
Beautiful. Beautifully
and
said.
I think we should push for it for the International Decade. Like, if they do nothing else, like, they've got 1.5 years to do something about this.
No, I really appreciate you bringing that to light too. For folks on the line. So just letting folks know we still have a short period of time with Dr. Daniel. So if you do have any questions, please do insert them. I have one other question that comes to mind, Dr. Daniel, for our folks who are living with sickle cell disease. Are there any considerations they should be taking given their, a medical minority already? Is there anything that they should be thinking about, not propensity, like you mentioned, for developing it, but anything you would like to let them know?
Again, I think it goes back to that. We need more representation. We need more physicians. Like, I don't know how many Black physicians in Ontario, Canada, are studying sickle cell disease.
Right.
And it's similar to malaria. It's almost like, if it's not impacting, regrettably, the white population, it's not a priority. Right. And so this is where we need, like, I know at McMaster, we have a physician who studies sickle cell disease, and she's white, and she is constantly frustrated that she cannot get various medical organizations to take it seriously. And this is a white ally who's been doing sickle cell research for 10, 15 years, and she's incredibly frustrated. So she's a great ally. She's mentoring many of our Black students who want to be doctors, because I think as well, there are not that many Black doctors studying sickle cell disease. Right. So she's been a great ally and mentor to many of our Black undergraduate students considering careers in medicine. So we need people like her, but we also need our healthcare system to realize that they have to do, like, some of these diseases are so easily treatable, or at least they're able to extend the lifespan of the patients if they just put a little financial investment and effort into. So I'm super happy to hear about that initiative you told me regarding scholarships, because, I think, like, that could be one that where we say, look, we want to have students that are going to study these diseases that really impact Black communities the most. So it's not now just about being a Black student per se, but actually you're going to start studying these diseases, whether it's sickle cell or malaria or whatever that's impacting Black communities globally, not just in Canada, but globally. Because if we're not doing it, no one else is going to do it.
No one's going to do it. You said it best. I mean, I want you to pick up on that thread about, overcoming the challenges of the system not taking these diseases or these discoveries seriously enough. How do you kind of navigate those within your own work and trying to bring attention to the KISO gene?
Well, to be honest, like people are like, a lot of my trainees regrettably did not stay in academia because they saw how I was treated.
Okay.
When I first started this research on triple negative breast cancer in Black women, my very, very first Canadian grant application to study this was rejected. And one of the comments from the reviewers was, "This is not relevant to the Canadian context." And that was in 2015.
This is recent.
Sorry, no, that was 2013.
Oh, okay.
Because I'm persistent, I reapplied the next year, still didn't get it, reapplied the next year, and I got it in 2015.
Okay.
It took me 3 tries to get funding for this research. Now, everyone's like, "Oh my goodness, Juliet, triple negative breast cancer, wow." And I'm thinking,
I've been saying this for a while. Yeah,
right? Like, so imagine if we had started this earlier, right? Like, it's just this idea that only certain people's ideas are valued or prioritized, or certain people are considered smart.
Yeah.
Since we're recording, I have to watch my words.
Yes.
Right?
Oh,
like I'm
man.
like, no. Every voice matters. And I say this to some of my friends, even like something as simple as our healthcare system is like about to implode. We have all these gifted immigrants here.
Yes.
Super trained physicians and dentists and lawyers in their home countries.
Yes.
We just need to develop a certification process. Even if it's 6 months, you can charge them 5,000, 10,000, whatever.
Right.
But they have the basic skills already. They just need to be certified with Canadian experience. Like, that's not rocket science.
But I mean, it is
if
a bit
we get sick in any of those countries, they're the ones that treat us.
Literally, exactly. You're not going to get airlifted back to Canada. So
right? So
exactly.
I'm like, help me understand why this is a problem. They're not taking jobs from Canadians. They're not.
No.
We need more healthcare professionals. We need more engineers. We need more people. Canada's a huge country and we don't have enough people in our workforce. And our workforce is demographically an inverted pyramid with way more people over 55 now than we have under 55.
Yes.
We need workers.
So I'm hearing some feedback from our audience members. One person, Raquel, mentioned that one of the biggest barriers is finding a supervisor for Black health in the program you want. So to your point, there's a dearth of just Black professors or Black doctors.
Yes.
And she says she's experienced consistently being streamlined into non-kind of science background, just given that I guess there's this, like you mentioned, this understanding or belief system that we're not capable of.
Exactly. And the majority of Black Canadian academics are in the social sciences because historically so the majority, like if there's 200 Black professors in Canada, there's more than that, at least 150 are in social sciences and humanities.
Yeah.
Right.
And could you remind me again of that network, the Black Canadian Scientists
Network?
So
it's the Canadian Black Scientists Network, CBSN. Okay. So we were co-founded in 2020, and like many other organizations after George Floyd's murder. Many of us knew each other before, and we had talked about doing something like that, but we never had the time, like literally. But then with George Floyd, and it was the middle of the pandemic, and we channeled our energy into forming this organization. But we've been doing it as well as a volunteer activity on the side of our desk. We were all completely burnt out by this. And we have to now constantly write grants, beg the university presidents for money to have a virtual conference. So we had our first conference in February this year. It was a huge success.
Fantastic.
And we had, over 1,500 people register to attend the whole conference. But then on any one day, we had 500 or so Black scientists or scientists to be attending. it was incredibly powerful. Many people were writing in the chat, they've never seen so many Black scientists in one space.
Right.
Many people commented on the fact that they left science because of how they were treated. Like it was a powerful, powerful event for everyone that attended. And, you know, we're doing it again in February 23, but again, we're all stretched thin. And here we are in December, and we're literally now trying to beg for donations and money to host this thing again. And you're saying, why?
Yeah.
Right? None of this is going into our pockets. This is like, it's, you know, and so these are the kind of things that are incredibly annoying and frustrating, but we're all incredibly passionate because we've all trained and seen our Black students lose hope and give up. And so there's many other initiatives I'm involved in, and that's partly why I'm tired all the time. I'm like, you know, like,
right.
I can give a talk on just all the initiatives I'm doing to increase the numbers of Black success in Canada, period. So it's yeah, I mean, if that's something public health can do and advocate for, you know, 3 years of funding for the Black Scientists Network to do this conference, because we cannot do this every year. We cannot spend time begging for money. Like, you know, you have to write the letters, you have to mail the letters, you have to follow up when they don't reply. Like all of that stuff I have to do this weekend, in addition to reading grants for a panel, a federal panel that I'm on. Like,
right. No good deed goes unpunished. It sounds like you're just, like you mentioned, Juliet, I mean, there's so few of you, you're getting stretched thin. And you're a trailblazer, and we appreciate you, applaud you for all you're doing, but there's only so much you can do as one person.
Exactly. So I think if the public health agency if you can lobby them and get them to, you know, like whoever, you know, and ultimately we need allies. We need allies, white, purple, or pink, to be advocating for us, because sometimes those allies come in unusual shapes and sizes. But we need someone to really say, listen, and again, we need someone to do the economics and show how this is, in the long run, this is hurting Canada's economy.
Yeah, big time.
Like we can't be just thinking, oh, everything should be based on merit, we're going to give it to the best candidate. Like,
and even that whole philosophy about who determines what's merit and who determines what success looks like and how we arrive at that is so skewed. You're right.
But also, like, some people aren't, you know, at that considered meritorious because they were not given access to begin with.
There you have it.
So I keep telling people, if you had 2 kids and you fed one a lot and you starved the other, you can't now complain and say the skinny, malnourished one could never win the 100-meter sprint. Of course they can't, because you never gave them food.
Exactly.
Not because they're not able to.
You presented so many barriers and unearned disadvantages. What do you expect? Got it.
So Anyway.
One person asked about the conference that you're hosting in February. Is there any way we could send them a link or?
I can send that. Let's see. I can probably even put it in the chat. Give me 1 minute. Can they see the chat though?
Yes. As long as you send it to everyone, they'll be able to see. There's a little drop down.
Let's see. There was just an email from our president reminding us. That's the abstract submission. I don't know if they want to submit to the attend or just to,
whatever you can in terms of promotional material would be fantastic, just so we can at least
I can just email it to you, and then you can share it to your whole network. That probably would be easier. Right? But it's from February 1st to 4th. It's virtual. It's free for Black identifying scientists and students. If, however, you're part of an organization or come in as an individual, we ask just for a little, like $50, just because, as I said, we literally have zero
funding.
You know, last year we were able to get, I think, 15 university presidents across Canada to donate, you know, like $10,000 to $20,000 each to run it. But this year, because every university is kind of having a little shortfall because of the pandemic and other issues, it's been a bit challenging to get, like right now I've only had responses from 3 universities out of the 12 that I sent sponsorship requests to. You know, it's a lot of work.
I definitely know Miss Landry is one of the trailblazers on our end who does a lot. Her days are dedicated to just that. So I know it's not for the weak of heart. So I'm not seeing any additional questions from folks. So maybe a last call, if there are any last closing thoughts or any questions that we wanted to pose to Dr. Daniel. Otherwise, we will wrap the session up and give you all back 35 minutes of your day.
And the sun's coming out here. Hope it's coming out in Toronto as
It
well.
is, it is, it is. I'm in Ajax. I'm seeing some
Yeah,
sunshine,
yeah,
flowing
yeah,
through. So I'm
yeah,
appreciating that.
yeah.
Okay, so I'm not seeing anything come through. So just wanted to let everyone know, thank you so much. There's a short survey we're hoping that folks can participate in, just letting us know how best we can continue to
Mhm,
deliver these sessions.
sure.
I've posted a link in the chat for you all. And then I'm just gonna share my screen one last time just to say thank you. We look forward to seeing you participate at future events that we have like this. And we hope that you're able to learn something and take away something from this experience. So with that, thank you again, Dr. Daniel, for joining us this morning.
You're
We were so
welcome.
fortunate and happy to have had you, and to all of you that participated and had your full attention throughout the entire 1.5 hours. Thank you, and we look forward to seeing you at future sessions.
Thank you so much and good luck and keep up the work you're doing.
Will
And
do.
yeah, and also send me the info about, like, I know you have an annual sickle cell like
Yeah, summit.
event and gala and fundraiser, like, so yeah, just add me to your mailing list so that I know when you're having those
Oh,
events. I can drag out some of my friends.
I'm definitely gonna take you up on that. Definitely will do.
And we need
So
to touch base as well about the scholarship and other things.
yes,
So yeah,
please, please, please. I'll send you a note after this
yeah,
conversation, but thank you so
yeah.
much, Dr. Daniel. Enjoy
No
the rest of your
problem.
Saturday.
Thank you.
All right.
Bye.
Take care.